Good day, everyone. For today, we will discuss genus Bordetella. So this is a figure showing the summary of the characteristics of Bordetella genus and the different species under this genus, which are the Bordetella bronchoseptica, avium, pertussis, and parapertussis, which we will discuss later in the subsequent slides. For the characteristics of the genus, in terms of cellular or morphological characteristics, the Bordetella are known to be small gram-negative rods or cocobacillary in form. They are motile. For the cultural characteristics, they are strict aerobic bacteria, non-fermenting. So they are non-fermentative in carbohydrates. They utilize amino acids for energy, and they can grow on non-enriched or ordinary media in McConkey agar. For the biochemical characteristics, they give positive results for catalase, urease, and oxidase tests. The usual habitat, they are known to be obligate parasite in the upper respiratory tract of animals. So they have an affinity for ciliated respiratory epithelium. They are opportunistic invaders, and they are labile in the environment. Survivability is less than few days, in few days only, so they can be killed easily. Differentiation of Bordetella bronchoseptica and avium, which are known to be the most important species of Bordetella affecting animals. They are differentiated based on their growth characteristics, biochemical reactions, their unique ability to agglutinate red blood cells. Bordetella avium requires differentiation from Alcaligenus fecalis, which is a non-pathogenic bacteria. For their growth on blood agar and MCA, for blood agar plate, the colonies of virulent strains are small, convex, and smooth colonies. And in blood agar plate, only the Bordetella bronchoseptica is known to be hemolytic, while Bordetella avium is non-hemolytic. Their growth in McConkey agar, they are colorless and pale because they are non-lactose fermenting, because they produce non-lactose fermenting colonies, both Bordetella bronchoseptica and avium. A selective indicator medium containing bromothymol blue as pH indicator can be used to differentiate Bordetella bronchoseptica and avium. There are available miniaturized biochemical identification systems for differentiation of non-fermenting bacteria, which do not metabolize carbohydrates. As we have mentioned, they are non-fermentative in carbohydrates. And hemagglutination can be used to determine the virulent isolates of both bronchoseptica and avium. So this figure shows the hemagglutination reaction, but this one is demonstrated using a virus. First, or the top panel shows the positive reaction of a hemagglutinating virus. When combined with the red blood cells, it can cause clamping or hemagglutination, while the bottom picture shows a negative reaction or non-agglutinating virus. So this table shows the different species of Bordetella with their disease conditions and the host they infect. For the Bordetella bronchoseptica, it can affect various hosts, and the most important hosts of this organism are dogs and pigs. In dogs, the disease is known to be kennel cough, or also known as canine infectious tracheobronchitis. So it can cause bronchopneumonia secondary to canine distemper disease, which is known to cause by a virus in dogs. In pigs, it can cause non-progressive atrophic rhinitis, together with the toxigenic strain of Pasteurella multocida that is known to cause severe progressive atrophic rhinitis. In cats, especially among kittens. There is pneumonia produced secondary to viral infection. In horses, the, the organism is known to cause bronchopneumonia after anesthesia, stress, and penicillin therapy. In laboratory rodents or among laboratory rodents, there is also bronchopneumonia, upper respiratory infections, and septicemia. For Bordetella avium, it can cause torticolliza, and pertussis and parapertussis are known to cause whooping cough, which is a highly contagious respiratory tract infection in humans. And in lambs, there is pneumonia for parapertussis, and Hinze is known to cause infection in immunocompromised humans, and it was also isolated among poultry species. This table shows the different features, characteristics of Bordetella bronchoseptica, Bordetella avium, and alkaline genus fecalis in terms of their ability to cause hemolysis in sheep blood agar. Only Bordetella bronchoseptica is hemolytic, while avium and alkaline genus fecalis are non-hemolytic. In McConkey agar, they have pale colonies. In terms of oxidase and catalase production, they are positive. The three are positive. Urease production, only Bordetella bronchoseptica is positive. Utilization of carbon exclusively for citrate, they are all positive. Malonate, only alkaligenous fecalis is positive. Nitrate reduction, only Bordetella bronchoseptica. They are all motile with flagella. Hemagglutinating activity of virulent strains. For Bordetella bronchoseptica, it is known to agglutinate RBC or red blood cell of sheep and cattle or ovine and bovine, while avium is known to cause red blood cell agglutination of guinea pigs. So we will start with the disease produced by the Bordetella bronchoseptica. So this organism is a commensal bacteria in the respiratory tract, upper respiratory tract of dogs, cats, swine, rabbits, horses, guinea pigs, rats, and possibly other animals. Infection can be endogenous, meaning the infection can originate from within. When the organism gain an or there is an opportunity to cause disease, like in the presence of other viral infection. So the organism can be an opportunistic pathogen, or the infection can be exogenous when it is acquired from outside source. And the organism is known to possess pili and flagella. The pathogenic attributes of Bordetella bronchoseptica. Number one is the fimbrial antigen that facilitates the attachment of the organism to cilia of the respiratory tract. It has also endotoxin that attracts neutrophils to the respiratory epithelium. Another is the adenylate cyclase toxin that is capable of altering cellular function of the host, including inhibition of chemotaxis, phagocytosis, and intracellular killing. A dermonecrotic toxin is also released by the organism that is responsible for the production of atrophy of the nasal turbinates. That's why it is associated in the non-progressive atrophic rhinitis condition in piglets and experimentally in rabbits, rats, and mice. The toxin is known to impair the ability of the osteoblast to differentiate, that leads now to atrophy of the turbinate bones in pigs. And a hemolysin is also being produced by the organism, which is responsible for the hemolytic effect. Hemagglutinin is responsible for the attachment of the organism to the respiratory cells. And the tracheal cytotoxin is responsible for the ciliostasis. Ciliostasis is paralysis of the ciliary apparatus in pigs and the extrusion of the ciliated epithelium. So this is a table that shows the summary of the different virulence factors of Bordetella bronchoseptica and Bordetella avium. So the hemagglutinin activity is known to bind to cilia. This is only present for Bordetella bronchoseptica. Pertactin also has same activity with hemagglutinin. It binds to cilia. Both the organisms have this pertactin. Fimbriae, this is also both present in bronchoseptica and avium, which is known to mediate the attachment of the organisms to the cells. The adenylate cyclase hemolysin is known to interfere with the phagocytic cell function, known to be present only or produced by Bordetella bronchoseptica. Tracheal cytotoxin is present both for avium and bronchoseptica, and its mechanism of action is inhibition of the ciliary action and kills ciliated cells. Dermonecrotoxic toxin or dermonecrotic toxin, it induces skin necrosis and impairment in the osteogenesis. That's why this one is responsible in the atrophy of the turbinate bones in pigs, and it is produced both by the organisms avium and bronchoseptica. Same with osteotoxin, which is toxic for osteoblasts, produced by the two organisms, and the LPS, which is known to stimulate cytokine release responsible in the inflammatory reaction. How it is transmitted? So it is transmitted via droplet inhalation, via aerosols, direct and indirect contact, and also the role of fomites, transfer on footwear or clothing, and on contaminated feeding utensils. For the pathogenesis, the organism must attach to the cilia of the respiratory tract, wherein it is known to cause ciliostasis or paralysis of the ciliary mucociliary apparatus that is known when the organism grow and colonize on ciliated tissues, followed by invasion of neutrophils. So there is inflammatory cell migration, especially of neutrophils, on the intact respiratory mucosa. How do we diagnose the organism? So the diagnostic procedures used, we can submit different samples or specimen for laboratory examination, like nasal swabs, tracheal aspirates, and exudates. The organism can be cultured in blood agar plate and McConkey agar aerobically at 37 degrees Celsius for 24 to 48 hours. The criteria for identification of isolates, their colonial appearance on blood agar or selective media, they both grow on McConkey agar, but they are not non-lactose fermenters. Colonies appear as pale, colorless, and biochemical profile can be used, slight hemagglutination test. So the disease is the disease produced by Bordetella bronchoseptica is known as the canine infectious tracheobronchitis or the kennel cough. Most important pathogen that causes kennel cough are also some viral agents, but the main pathogen is the Bordetella bronchoseptica. But the role of other viral agents like the canine adenovirus 2, canine parainfluenza virus 2 play also an important role in the pathogenesis or in resulting more severe infection in dogs. So this is one of the most prevalent respiratory complexes of dogs. Other microbial pathogens that are also implicated in causing canine infectious tracheobronchitis are canine distemper virus, canine adenovirus, canine herpes virus 2, and reoviruses 1, 2, and 3, and other mycoplasmal species. For the transmission, via respiratory secretions, either by direct contact or by aerosols. Mechanical transfer on footwear or clothing on contaminated feeding utensils and yung mga role ng mga fomites are also known, which was mentioned a while ago. Morbidity rates can be 50%, and mortality rates is known to be low. For the clinical signs develop within 3 to 4 days after the exposure of the animal, of the dog. There is coughing, gagging or retching. Mild serous oculonasal discharge can also be observed. Affected dogs remain active, alert, and non-febrile. So there is no fever observed among dogs affected, and the disease is self-limiting. When you say self-limiting, even if you do not give antibiotics, the dog can recover. But of course, when it is in combination with other pathogen that we mentioned, like mycoplasma, other viral agents, adenovirus, para-influenza, so you need to give antibiotic treatment. How do we diagnose? Based on a history of recent exposure to carrier dogs. Based on the clinical signs, the gagging, the coughing, retching, signs that can be seen, transtracheal aspiration fluid is appropriate specimen for laboratory examination. Hemagglutination of ovine and bovine red blood cells among virulent strains of Bordetella bronchoseptica. Serology can be used in association with vaccination history. It has value in determining involvement of other respiratory viruses. So for the treatment, so since we mentioned the condition is self-limiting, those dogs that have mild clinical signs do not require specific therapy. Antibiotic therapy is usually recommended if coughing persists for more than 2 weeks or if bronchopneumonia is already present. So among the antibiotics that we can use are amoxicillin, tetracyclines, and fluoroquinolones like enrofloxacin. So other drugs or antibiotics that can be used are carbonicillin, chloramphenicol, tetracycline that we mentioned, thylacine, ceftiofur, sulfametazine, erythromycin, aminoglycosides, and polymyxin B. So the organism is known to be resistant to penicillin. For the control measures, isolation of the affected dogs immediately, correct the predisposing factors or the contributing factors when identified. There is a vaccine commercially available to protect the dogs from this kennel cough or infectious canine tracheobronchitis. One is the intranasal vaccine containing Bordetella bronchoseptica and para-influenza 2 antigen. And other kennel cough vaccines are being given subcutaneously, just like other vaccines in dogs, like rabies and 5-in-1 or 6-in-1 vaccines. But intranasal vaccine is also present. So they are usually modified live vaccines or MLV vaccines. So since the bacteria Bordetella bronchoseptica is also known to cause trophic rhinitis in combination with other organisms like the Pastorella multocida type D. So the toxigenic strain of Bordetella bronchoseptica can cause turbinate hypoplasia without distortion of the snout in young piglets under 4 weeks of age. Infection with this organism may facilitate colonization by Pastorella multocida type D, with the subsequent development now of severe atrophic rhinitis and distortion of the snout. So next we have Bordetella avium that is known to cause the condition turkey coryza or avian bordetellosis, an economically important disease of young turkeys and quail. So this is an acute rhinotracheitis with a high morbidity but low mortality, just like the Bordetella bronchoseptica infection in dogs, there is only low mortality. Contributing factors that leads to more severe infections are the presence of various stresses and other secondary infections caused by viruses like Newcastle disease virus and bacterial infection like the one that is produced by Escherichia coli or E. coli. How is the transmission or the modes of transmission of Bordetella avium? By direct contact, by aerosols, and by other environmental sources. The clinical signs observed are oculonasal discharge, coughing, sneezing, and dyspnea with decreased weight gain among turkeys. There is excessive lacrimation observed, and the turkeys usually breathe on their beaks. In severe cases, the tracheal cartridge is distorted and discolored, and there is collapse of the trachea that leads now or result to suffocation. What are the pathogenic attributes or the virulence factors of Bordetella avium? A is the dermonecrotic toxin, which is known to be present also for Bordetella bronchoseptica. Tracheal cytotoxin cause deciliation and loss of mucous gland function of respiratory epithelium. The presence of pili and hemagglutinin, which are known to help the organism in their adherence to the tracheal epithelium. How do we diagnose? Via clinical signs and gross pathological features. Isolation and identification of the organism from sinus and tracheal exudates. Virulent isolates agglutinate guinea pig red blood cells, which is in contrast with Bordetella bronchoseptica that is known to agglutinate red blood cells of sheep and cattle. Microagglutination and ELISA techniques can also be used, and some PCR-based methods can also be employed for the isolation and confirmation of the organism. For treatment and control, there is an available vaccine. This is a live attenuated vaccine for Bordetella avium. MLV or modified live vaccine can also be used in susceptible flux. The use of broad-spectrum antibiotics like tetracyclines to prevent or treat secondary colibacillosis, which is caused by E. coli, can be helpful. Thorough cleaning and disinfection of turkey houses after an outbreak is recommended. So that ends the genus Bordetella.